Hi, I am Brian Miller, CEO and co-founder of Sentien Biotechnologies. We are a cell therapy company developing a technology that restores balance to the immune system. Our lead program is SBI 101 for the treatment of systemic inflammation. For those of you that are familiar with us, I do have some updates that I'm excited to share with you this year from past events at Mesa. But as an overall review of our technology, we are an ex vivo cell therapy company treating systemic immune-mediated inflammation. The real innovation behind our approach, we are using mesenchymal stromal cells, allogeneic source from the bone marrow. Our specific innovation is how we administer those MSCs. So instead of injecting the cells into the body, either IV or into the tissue bed directly in some form or topical application, we are administering them ex vivo, meaning that we are putting the MSCs into a hollow fiber device on the outside of the hollow fibers where blood flows through the inside. And what that allows for is bidirectional communication between the blood component and the cellular compartment, where you can have molecular but not cellular interaction. By doing it this way, we believe we have greater control over therapeutic exposure than other forms of administration. And we are a clinical stage venture-backed company. We do have data that I'll share with you in dialysis requiring acute kidney injury that is overall supportive of anti-inflammatory and immunomodulatory effects that are commonly seen and hypothesized with MSC therapies. We are advancing into clinical studies for severe COVID-19, which I'll describe a little bit more as well. We have an open IND, and we'll be initiating our first study for severe COVID-19 patients in Q3 of this year, i.e. the next month or two. One of the most significant updates is we've added 2 members to our team. We have Rich Gans, who has joined as executive chairman. He has a long history in critical care applications, as well as the renal business. And then Dr. Alan Nissenson has joined us as well as our chief medical officer. He also is well renowned in the renal industry, as AKI is our first indication, with experience most recently as chief medical officer at DaVita. So we're very excited to have both Rich and Alan on the team. Others of us you may have met in years gone by, myself, Bijay Parikadhan, the scientific co-founder of Sentien, and then Chris Jamiti and Rita Barcia on our R&D and operations team. So the indication that we're treating is really a biologically inspired syndrome, more so than a specific indication. And this is really around the idea of systemic inflammation. When you have primary tissue injury, you get both immediate pro-inflammatory response, and there's also a compensatory anti-inflammatory response. And it's the balance of these two that really defines what recovery looks like. So the immune system is orchestrating this inflammatory response to injury. And in the good setting, you'll have normal recovery with a balance of these two responses over time. What can happen, though, with an acute injury is you can have a dysregulation of this immune system, an overreactive immune response, very significant primary tissue injury, and that can lead to certainly mortality as well as long-term morbidity if not resolved, and getting the immune system and the inflammatory response back to homeostasis. So the idea behind our technology is really to create this therapeutic intervention that orchestrates this return to homeostasis, this rebalancing of the immune system back to a normal recovery type level. And so the data we'll show you from the clinic, we believe supports what we're trying to convey here with this approach of rebalancing the immune system and getting back to homeostasis. As all of you know, with COVID-19, there is a severe hyperinflammatory response that when experienced in severe patients with COVID-19 really contributes and exacerbates the injury, the primary infection. So it's really getting this host inflammatory response under control that really for patients that are further along and more severe will dictate their prognosis, even if the viral aspect of disease has been well managed. So it's there that many people, including many of you viewing this presentation, understand that MSC therapy is well suited to address this systemic inflammatory response, this overreactive response. And why is that? It's really because MSCs are known to have this broad-acting characteristic where they secrete multitudes of cytokines that collectively lead to this tissue repair, regenerative, anti-inflammatory response. And it's really this complex pharmacology that enables treatment of these very complex pathophysiologies. In addition, MSCs, being a living cell, can respond dynamically in a microenvironment in which it finds itself. And this is obviously very, very important in the context of patients that are heterogeneous at baseline, but also dynamic in their clinical presentation. So it's really this combination of having broad-acting mechanisms of action, as well as a dynamically responsive, almost a self-tuning of MSC therapy to the patient's blood that we think really are the key benefits of MSC therapy. We believe that administration through direct injection limits the exposure of what you can get from these MSC secreted factors when you're infusing them IV or even directly injecting the tissue bed. You're not getting the systemic type of exposure that you would hope for. So what SBI-101 sets out to do is solve those two problems specifically. And as I mentioned earlier, we do this by inoculating the MSCs into a hollow fiber device. Instead of bringing the cells into the blood, we like to say that we bring the blood to the cells. So how this is administered is in the context of this extracorporeal blood conditioning type treatment, where blood flows through SBI-101, again, enabling that communication between the MSCs and the blood compartment. So pro-inflammatory mediators from the blood will potentiate MSCs and activate this dynamic response. So you really maintain the key benefits of MSC therapy, i.e. this broad-acting, dynamically responsive characteristics, while also enabling extended exposure and greater therapeutic overall exposure by incorporating the MSCs in SBI-101 in this way. As an added benefit, we can actually measure the pharmacology from the device during treatment. So that allows us to characterize how the MSCs are behaving in the context of each individual patient. And lo and behold, the data that we have from our first study shows that the MSCs do, in fact, respond uniquely to the unique inflammatory signatures of the patient. So we think this is really providing the initial evidence that we are maintaining these benefits of MSCs, this broad-acting, dynamically responsive characteristics, while also getting the longer duration of exposure. So the way this is clinically incorporated is in the context of existing blood treatment modalities. So for example, dialysis. We started our clinical development in acute kidney injury requiring dialysis, where SBI-101 can simply be integrated as an add-on to existing treatment. So on one hand, they'll be getting their standard dialysis treatment that they would normally get, but in addition, they're also getting SBI-101 treatment. So you're giving renal support in real time while also addressing the inflammatory response that is driving a lot of the pathology in acute kidney injury. This can be extended obviously into other tissue types, other treatment platform modalities like ECMO, photodynamic therapy, plasmapheresis are common examples of blood conditioning treatments. SBI-101 can be administered in these contexts or as a standalone hemoperfusion treatment where you have a blood circuit with just SBI-101. We're pursuing each of these approaches in ICU settings. So the clinical data that we generated in our phase 1b study in acute kidney injury really to again provide support with a little bit of data in this presentation around how SBI-101 is working. What we're showing here is a collection of 150 to 200 cytokines that were input into a systems biology tool called Ingenuity Pathway Analysis. What this tool does is it describes the pathways that are either being activated or inhibited based on the cytokine profile. And when we compare SBI-101 to sham, and this evaluation was taken days after SBI-101 treatment was completed, shows this broad inhibition of inflammatory pathways such as maturation, activation of lymphocytes, inflammation of the organ, other types of inflammatory mediated pathways are all being broadly inhibited by SBI-101. So this is really, again, showing that idea of this broad acting treatment that's really needed for these complex diseases. On the sham side, by contrast, you're seeing these pathways remaining activated relative to the SBI-101 treatment. The one example that was unique here, which we have with an asterisk, is a patient that was undergoing resolving sepsis. So she had sepsis when she was randomized into the study. She then was going through a natural recovery. So her sepsis and her inflammatory processes were resolving. And lo and behold, her profile of a natural resolution of inflammation looks similar to what SBI 101 is addressing in these patients. So we found this to be a very powerful representation of how we are trying to accelerate and improve this healing process and this normal return to homeostasis, as evidenced by, again, very small numbers, but this compelling initial evidence. Looking at the tissue level, this is a cartoon showing the renal tissue. And as you might expect, pro-inflammatory mediators like interferon gamma, TNF-alpha show up in the early injury phases. M1 macrophages, Th1 cells are all involved in the early injury process. But you would also expect to see later on a resolution in the form of IL-10, TGF-beta, M2, and Th2 cells. All of these things are, again, sort of classic wound healing. When comparing SBI 101 to sham, we showed a reduction in our treated patients of several of these early injury markers, and we've included KIM1 as a renal-specific injury marker relative to sham. So SBI 101 was reducing the injury markers and was upregulating the regenerative and healing markers. So again, this supports the notion of this therapeutic hypothesis where we are trying to accelerate these patients out of the injury phase and into the wound healing regenerative phase to try and get them back to homeostasis and on the road to recovery. So even in this small data set, we were very, very encouraged by what we were seeing. There was a nice spread. We're happy to show you more data under confidentiality, but this was a very good start for us. As we translate this into the COVID-19 hypothesis, we have more data, again, behind this, but just as a teaser, the idea of systemic inflammation in COVID-19, while there may be some unique characteristics, what we saw in non-COVID AKI patients would be supportive of the types of changes you would hope to see in COVID-19 patients as well. So we believe that the biology observed in our non-COVID AKI patients really, again, overall supports this translation into COVID-19. Not that we're going to pivot to be a COVID-19 company specifically, but there's a unique moment in time for us to demonstrate how this product can address systemic inflammation in the context of this pandemic. So in terms of our overall company pipeline, as I mentioned, we are pursuing COVID-19 patients with AKI-D, so we're taking the combination of the work that we've done and applying those learnings into the COVID-19 setting. So we're going to be activating that trial here very shortly. We will also be initiating a study very soon in ARDS only, so these patients would be less severe than those that are also requiring dialysis. We hope to initiate that in the second half of this year as well. And in the meantime, we will continue our work in advancing other critical care applications where systemic inflammation is known to be an issue. Acute liver failure, burns, trauma, these are all critical care-wide type indications where systemic inflammation is known to play an important role in the prognosis for these patients. Last but not least, we have begun work on chronic applications as well, where we could think about incorporating with either outpatient dialysis or plasmapheresis type settings in both the autoimmune setting as well as neuroinflammatory disorders. So that work is earlier, but again, we are very encouraged by what we've been seeing on our preclinical in vitro platform, characterizing patient blood in the context of these indications. So we're going to continue that work, obviously, as well. In summary, I hope to convey to you today that SBI 101 is designed to treat systemic inflammation in the presence of tissue injury. We're initially focused in the acute critical care realm, and we do this by extending exposure to MSC secreted factors, which leads to this idea of rebalancing and reprogramming the immune system back to a normal homeostasis and recovery and a tissue repair type response. The clinical data that we've generated so far supports this therapeutic hypothesis. We are very motivated and encouraged by what we've seen. So next steps are for us to test this initially in a pilot phase study with COVID-19, but then extending that through proof of concept. We really want to show that this reduction of systemic inflammation will then translate into improved clinical outcomes. And obviously what we need to do is treat more patients in order to demonstrate that. Along with that, continue development in non-COVID acute inflammatory diseases. As the pandemic, hopefully in the next year, begins to recede, that clinical sites will be more open for business on some of these non-COVID applications, which remain a very important need and continues to support the therapeutic and business hypothesis of SBI 101 for the long term, as well as extending our platform for chronic indications. So I thank you all for your time. I'm sorry this year we can't take questions. I will just say that, again, that the team is very excited by where we are, and we're looking forward to continuing this important work. Thank you for your time.